Part 2, Immunology. Immunology means that immune is the immune system, logy science, so it is science of protection. In this chapter we will talk about the role and components of the immune system, what are the equipments are used in the first chapter, what are the equipments that are used for fighting against any invasion and protecting our body. And in the second chapter we will talk about the mechanism the immune system undergoes to protect our body from any invasion. In the last chapter we will talk about immune diseases. So let's start with chapter 6, role and components of the immune system. Immune system is present in our body to protect it from bacterial or other invasions that may cause diseases. The immune system is made up of two main parts: the immune cells, represented by the white blood cells, leukocytes, example, granulocytes, lymphocytes, mastocytes, etc. Immune molecules are represented by antibodies and complements. Document 1, HLA, a major self-marker. The white blood cells are immune cells responsible for elimination of any invader, pathogen, that cause disease. In 1 millimeter cube of blood, there is 5,000 white blood cells are present. The immune system, our immune system, is able to differentiate between 3 types of antigens: self-antigen, that is a part in our body and doesn't cause any disease, example, bone cell. 2. Non-self antigen is any antigen that enters our body and causes disease, example, bacteria. And modified antigen is a self-antigen that transforms under certain genetic modifications, that is, under variations, mutation, like into non-self antigen, example, cancer cell. HLA by definition is a human leukocyte antigen which represents a biological identity card specific for each individual, where only identical twins have the same HLA. These HLA represent the self-markers of white blood cells. Those white blood cells and other nucleated cells of our body contains an identity card called HLA that protects itself from any responses done by immune system in order to prove for them that they are self-antigens or self-cells that are present as an element, as elements in our body. Okay. During transplantation, grafting, all scientists look for the antigens of the donor and the recipient, the HLA antigens. They are expressed, as we know, by a gene present on long arm of chromosome number 6, represented by 6 loci, which are DP, DQ, DR. Okay? Now you should make interpretation of document A. After interpretation, you must make a conclusion. Please make interpretation and then switch the sound in order um, to know the results. Now, after making interpretation, we can conclude that the acceptance or rejection of graft depends on compatibility of HLA between donor and recipient. There are four types of graft. Okay, do you know them? The types of graft are four: autograft, which is a graft in the same individual; isograft, which is a graft between two identical individuals from one individual to identical one, like twins; allograft, which is a graft between two different individuals of same species; and xenograft, which is a graft between two different individuals of different species. Okay. In document C, there is a sample of a part of chromosome number 6 long arm showing the organization of MHC in man. As we know that MHC is a major histocompatibility complex. This definition MHC is used as HLA, but since HLA and MHC are present in all species in nucleated cells of these species, we can differentiate MHC and HLA as that those for animal, and HLA the cells of a human, mainly white blood cells of a human. For that they are called human leukocyte antigens, that is antigens present on the membrane of white blood cells. Leukocytes mean white blood cells. Okay. The gene of HLA is represented by 6 loci, as we know. These loci are divided into two groups, class 1 and class 2. Class 1 antigen is present on class 1 antigens are expressed by 3 loci BCA. This BCA codes for protein called one polypeptide protein, which form a polypeptide chain that attach to the plasma membrane of the nucleated cells. Class 1 is present in all nucleated cells of the body. They represent the HLA antigens that are the self-markers which show the card for the immune system in order not to kill it. Okay. فإذا they are expressed by 3 genes, class 1 BCA, 3 loci BCA. They code for one polypeptide chain, and they are present in all nucleated cells. Okay, class 2 HLA is expressed by 3 loci DPDQDR, which codes for class 2 antigen having 2 polypeptide chains present on the plasma membrane of the immune cells. But as you know, immune cells are nucleated cells. Then, since they are nucleated cells, so they also contain class 1. For that, nucleated cells contain only class 1, while immune cells contain class 1 and class 2. That means that they have the 2 polypeptide chains of class 2 and the 1 polypeptide chain of class 1. Between class 1 and class 2 loci on the HLA of the HLA gene on chromosome number 6. Between them there is class 3 gene which codes for chemical molecules called complements. They have an effective role for the immune system. Later on we will know what are their role. Okay. In document B, they are asking for to make interpretation for the document B, which represents the percentage of transplant survival in two different studies, one in Eurotransplant and the other in Oxford. Showing the incompatibility number. Compatibility means similarity. Incompatibility number means difference. Incompatibility number between donor and recipient of HLA-A and B in Eurotransplant and for HLA-DR in Oxford. Okay, so look at the graph. On x-axis they represent the HLA-A and B for Eurotransplant in Europe and HLA-DR for the transplant on Oxford after 1 year survival of transplant. The percentage is represented in the given. Note, during interpretation, you must look when you are making analyze at the main points, okay? Not point by point. You will make then description. We want to interpret, analyze plus interpretation. Okay? Please make interpretation and look on the paper about the results. Okay? So, after you make interpretation, I can conclude that the survival of transplant depends on the incompatibility number between HLA-A, B, and DR. That is, transplant, as we mentioned in the notes previously, depends on compatibility of HLA between donor and recipient. Okay? Document 2. Document 2 is talking about blood groups. Blood groups, they are the self-markers of red blood cells, erythrocytes. As we mentioned previously, that the self-markers of white blood cells are called HLA. But the blood cells, red blood cells, doesn't have nucleus, so they need specific markers. The markers that represent antigens of red blood cells are called agglutinogens also. These self-markers are present on the membrane of red blood cells. Okay. These agglutinogens are made up of carbohydrate and protein. So they are made up of glycoproteins. Glyco from glycogen, okay? These agglutinogens are glycoproteins present on the membrane of red blood cells, which is made up of phospholipid bilayer, the membrane of red blood cells, attached together by carbohydrates and proteins called oligosaccharides, proteins. Okay? To determine the blood group, the following procedure can be done. At first, pick the tip of the finger with a sterilized lancet. Then, put separately 3 drops of blood samples on a glass slide. Add separately anti-A, anti-B, and anti-T on the above sample. Wait 5 minutes and observe where agglutination occurs. Okay? If agglutination occurs with anti-A, then the blood group is A. If agglutination occurs with anti-B, so blood group is B, and if agglutination occurs with anti-RH, then the individual is positive. If no agglutination occurs with anti-RH, then he is negative. Note, blood group O is a universal donor, while blood group AB is a universal recipient. How we know if blood transfusion is accepted or rejected? Okay, we look at the antibody of the recipient and the antigen of the donor. Here is a table of blood group. During the blood group A has on the membrane of red blood cell a self marker, which is the antigen A, and in the plasma antibodies called anti-B. Blood group B has on the membrane of red blood cells antigen B and antibodies in the plasma called anti-A. For blood group AB, there is no antibodies in the plasma, while antigen in the red blood cells are both A self-marker and B, represented equally in blood group AB. And lastly, a blood group O has antibodies in plasma called anti-A and anti-B, while antigen in red blood cells are not present. Okay? Keep the note in your brain that during blood transfusion, we look at the antigen of the donor and the antibody of the recipient. Okay? Then, solve the below exercises. Okay. Interpret document AB. Interpret document B. In document B, there is a table showing serum from different blood groups and the result after mixing the red blood cell of the given group and the serum of this group, and noticing the result, we notice that there is agglutination in some and no agglutination in others. Okay? Agglutination means accumulation of blood, means that the antigen and antibody bind together and cause agglutination blocking. تخثر يعني. Okay. Please make interpret, then see conclusion. Okay, we notice that each blood group has its antigen but not its antibody. That is, blood group A has antigen A but antibody B. Why? In order not to make agglutination between antigen A and antibody A. That is, if my blood group is A, then it has on membrane of red blood cells antigen A. If my serum has antibody A also, then antigen A plus antibody A in my serum will cause agglutination, that is, will bind together and cause agglutination, leading to fever, high temperature, intravascular lysis of blood vessel, and may cause death. Okay, so each blood group has antigen and not antibody of the same antigen. Okay, for that they should be opposite in order not to make agglutination and cause any side effects. This is blood group transfusion diagram. As we see that O is a universal donor, receive gives the blood to all, while AB is a universal recipient, which receive blood from all. Okay, and each blood group give herself blood transfusion. Okay, while A gives only AB and itself, while B gives also AB and itself only. RH transfusion. RH transfusion, we look at the antigen of the donor and the antibody of the recipient. Do you know what do we mean by RH? Okay. RH is a human antigen present on the membrane of red blood cells, similar as that antigen present on the membrane of red blood cells of rhesus monkey. That means that in addition to antigen A and antigen B, which represent the self-markers of the blood group, there is also additionally another antigen called RH. This antigen is called so since it is same as that present on the membrane of red blood cells of monkeys called rhesus monkeys. For that, these antigens are called rhesus antigen in reference to monkeys. Okay? A person which carries on the membrane of red blood cell RH antigen on the membrane of red blood cells, then this individual is called the rhesus positive individual. That is on the membrane of red blood cells is present as a self-marker. We can conclude that this individual doesn't have antibody RH. Okay? You know why? If antigen RH is present and also anti-RH is present in the same individual, then a blood transfusion the blood will coagulate and cause high fever and intravascular lysis. Also, if a person that is rhesus negative, which means that no rhesus on the membrane of red blood cells, then he may or may not has anti-RH in his serum. Why? As we know that the antibodies of the ABO system are acquired since birth. Example, any individual which is born newly, if we make blood analysis, we will see that if he example B positive, A negative. Okay, so we know if he is B positive individual, we notice we can say that he has antigen B and antigen RH. And we can notice that his serum will have only anti-A against B. Okay? If a person is A negative, then he has antigen A, but he has no antigen RH. In his serum, he will have antibody B, opposite as we said previously. So, the antibodies of ABO system are acquired congenitally since birth. Congenitally means since birth. However, the anti-RH are acquired naturally by accident. The RH group is expressed by a gene which doesn't form antibodies unless the negatively RH person is subjected to an accident causing the manufacture of antibody. Then the individual which is negative, he may have or may haven't anti-RH. Okay? During blood transfusion, the Rh transfusion, we notice that if the person is Rh positive, that is, he has antigen Rh, if he gives to recipient with Rh positive, the recipient doesn't have anti-Rh, so the blood transfusion is accepted. From Rh positive person which has antigen Rh to Rh negative person anti-Rh, the blood transfusion is rejected since antigen Rh will bind with anti-Rh and cause agglutination. From Rh negative to Rh positive is accepted since the Rh negatively person doesn't have antigen, while Rh positively person will not have anti-Rh also, for that they are accepted. And the blood transfusion from Rh negative to Rh negative is accepted since there is no antigen Rh in the donor. Okay? Now you should make interpretation of document F. Before making interpretation, you must read well the document, which represents the identification of rhesus group in document F. To simplify for you, as you see that the rhesus monkey, there is 3 subjects: rhesus monkey, rhesus positive individual, and rhesus negative. From the 3 subjects, RBCs are taken and placed on a glass slide. Then a serum from a given rabbit is taken, blood serum, and separated to the 3 samples. The result was negative. Why? You should know. For the another document in F2 يعني, الماكاكو monkey, rhesus monkey, has rhesus positive is together with rhesus positive and rhesus negative individual. Red blood samples are taken and placed on a glass slide as previously, but additionally, a given sample of RBCs from rhesus monkey was injected to the rabbit. Then, from the rabbit, an immune serum was taken and separated to the 3 samples, as shown in the glass slide. Agglutination occurs only with rhesus monkey and rhesus positive individual, while no agglutination occurs with rhesus negative individual. Interpret, and what can you conclude? Did you solve it? Okay, as a conclusion, we can notice that we can say that anti-Rh antibodies is acquired naturally and not congenitally. Okay? We should mention that rhesus is expressed by chromosome number 1.